Good morning. I am Dr. Yogesh, Professor and HOD, Department of Physiology, Sri Chamundeshwari Medical College Hospital and Research Institute, Channapatna. Okay, so now let us see the next experiment, that is the bleeding time. So in this video I am going to cover three things: bleeding time, clotting time and blood grouping. So in bleeding time you have to know the method. The method what we are using here is the Duke's method. Along with that, in all these experiments, know the definition. What is bleeding time? You have to know compulsorily. What is the normal time? And in which conditions the bleeding time is prolonged. These are must to know. Now another common mistake what you make during the procedure is the time starts as soon as you have pricked your finger, and whatever you are going to put the first blot, that is going to be the 0 seconds. Some of you make a mistake that you mark that as 15 seconds. No, the first blot is 0 seconds and you're subsequently blotting every 15 seconds till when you are going to do till the bleeding stops. Here also again we see many of you do a mistake telling that whenever there's a last blood drop, you mark that and you write that is the end. That is, how can that be the end? Because if you mark that as the end, when the blood is present over there, it means you are still bleeding. So that is not the end. End mark should be an empty circle. So this is another common mistake what you are going to do in bleeding time. Okay. So let's proceed with the clotting time. So in clotting time, the method what you are going to use is the capillary tube method. It is always good to know some additional one or two other methods which can be used even for the bleeding time. So clotting time, same thing, know the definition and know the normal clotting time, and you should know in which condition the clotting time is prolonged. Okay, the discussion will continue on whatever you are going to answer accordingly. And here also same thing. For example, let's say you are telling that clotting time is prolonged in hemophilia. Definitely they are going to ask you what is hemophilia, what factor deficiency is causing it. So it continues like that. So that is for the clotting time. Next is the blood grouping. When you get blood grouping, you know the procedure how to do. Here now again what you should compulsorily know is Landsteiner's law. So there are two parts of the law. You should answer both the parts, and why there are two parts, that also you should know the reason. Other common things what can happen during the discussion is they can ask you what is this anti-sera made of, in what are the different types of blood group systems, in which conditions, what is the use of doing this blood grouping, the hazards of blood transfusion, hazards of mismatched transfusion, what is cross matching. These are some of the commonest questions you have to concentrate and be thorough. Thank you.